Dr. Adina, can you hear me?
Sir, I can hear you.
I hope Adina can hear me as well.
Second him.
Adina, yeah, are you ready? I'll just do a brief introduction and then we can start.
Sorry, I cannot understand.
Are you ready, Adina?
Yes, sir, I'm ready.
So I welcome you all to this session called the Resident Corner module. We used to do these sessions before COVID, and the idea behind these sessions was to build community international for the residents. We are restarting these sessions, and these sessions will be there every Thursday. Today we're going to talk about white matter trash. Dr. Adina is A1 neurosurgeon at Yakin National Hospital. These sessions are arranged by the Department of Neurosurgery at Yakin National Hospital, and I urge everyone to join and make use of these sessions. Dr. Adina, can you start? And make sure you use a pointer while explaining.
Yes, sir. Good morning, everyone. Today I'm presenting white matter tracts of brain. So, okay, so gray matter is of the brain is made up of cell bodies, and whereas the white matter is composed of myelinated nerve fibers, the axons. So, white matter of the cerebrum underlies the outer lamina of gray matter, intervenes between the cortical gray matter and the gray matter of the basal ganglia and cases the ventricle. So, the white matter is underlying between the gray matter, cortical gray matter, and the basal ganglia, and it encases the ventricles. So there are three types of white matter tracts. They are association fibers, commissural fibers, and projection fibers. The association fibers are the fibers that are interconnecting the different cortical regions in the same hemisphere. So these are the association fibers, whereas the commissural fibers are the fibers that are interconnecting the two hemispheres across the median plane. And the projection fibers are the ones that are passing up and down the neuroaxis, connecting the cortex with caudal parts of the brain and the spinal cord. So there are three types of white matter tracts in the brain. So starting with the association fibers are again of two types: the short arcuate fibers that interconnect the adjacent gyri, and the long arcuate are the ones that interconnect widely separated gyri. So here we can see an example or a picture of a short arcuate fiber that is interconnecting the two gyri. So here we can also we can see that this is a sagittal cut of the midsection of the brain. Here we can see that the arcuate fibers are crossing from one gyri to the another gyri. The other tracts we will discuss later on. There is another type of association fiber bundle encountered in the ventricular margin is the stria terminalis that arises in the amygdala and courses along the border between the caudate nucleus and the thalamus in the wall of the lateral ventricle. So here we can see this is the sagittal section of brain. Here we can see that there are stria terminalis. These fibers are they arises from the amygdala and courses along the border between the caudate and the thalamus in the wall of the lateral ventricle. So the uncinate fasciculus is a type of association fibers. These are hook-shaped bundle of fibers, and they connect frontal and temporal lobes of the same cortex. Location is at the lateral edge of the anterior perforated substance, and it has two components: the lower component, the upper component. The lower component that connects the orbital surface of the frontal lobe with parahippocampal and other gyra of the medial surface of the temporal lobe. So it is the connection of the orbital surface of the frontal lobe with the medial surface of the temporal lobe. whereas the upper component, it connects the gyri on the superior lateral part of the frontal lobe with the lateral temporal gyri. So here, this is the sagittal section of midsection of the brain. The claustrum and posterior part of the external capsule has been removed to expose the putamen, which is the basal ganglia. The anterior part of the external capsule has been preserved. We can appreciate the uncinate fasciculus that is connecting the frontal lobe with the temporal lobe. So these are the fibers that are running from frontal to temporal lobe, and it is below the putamen. In this picture, we can see that the temporal horn, the body atrium, and frontal horn, temporal horn of the ventricle has been exposed. Here we have exposed the frontal horn, body, temporal horn, and temporal horn, the atrium of the lateral ventricle. The fibers of the external capsule superficial of the putamen has been removed, and the lower part of the uncinate fasciculus has been removed to expose the amygdala and the head of the hippocampus. So here, the lower part of the uncinate fasciculus has been removed, and only the upper part is present. That was connecting the frontal lobe to the medial temporal region. Another form of association fibers are the cingulum along the medial aspect. They are present along the medial aspect of the cerebral hemisphere. It contains long and short association fibers that follow the curve of the cingulate gyrus, carpus callosum, and forms much of the white matter of the cingulate gyrus. So what it connects the subcallosal and para-olfactory areas of the isthmus, and the isthmus of the cingulate sulcus and hippocampal gyri. So here we can see the sagittal section of the brain. Here we can see that this is the carpus callosum, and here is the cingulate gyrus. The cingulum is covered or is in between inside the cingulate gyrus. Here we can see the cingulum. As we have exposed the cingulate gyrus, we can appreciate the cingulum. The section extends through the medial part of the right hemisphere and thalamus. It crosses the medial part of the head of the caudate nucleus anteriorly and pulvinar of the thalamus posteriorly. The cingulum is the bundle of association fiber that wraps around the outer border of the carpus callosum. So here is the carpus callosum, and it wraps around the border of the carpus callosum. And and it is found the depth of the cingulate gyrus. So if we see, this is the carpus callosum, this is a cingulate gyrus. It is found in the depth. If we remove the cingulate gyrus, we will see the cingulum. Its connections are anteriorly and below. It is connected with the para-olfactory gyrus, and superiorly it is connected with the cingulate gyrus, and below and inferiorly it is communicating with the parahippocampal gyrus, the cingulum. Now the other tracts are the superior longitudinal fasciculus. They are located along the upper and lateral border of the lentiform nucleus and insula. So lateral to the, and it is found lateral to the internal capsule. It connects frontal, occipital, and temporal lobes. Here, the frontal, parietal, and temporal operculi has been removed to expose the insula. This is again the sagittal section, and the superior longitudinal fasciculus actually courses from the frontal lobe. It goes behind, and it goes behind and below the parietal lobe, and it comes forward and to the temporal lobe. So here the superior longitudinal fasciculus courses around the outer margin of the insula and lentiform nucleus. So this is the insula, and we can see that these are the fibers crossing from frontal, occipital, and then the temporal lobes. In this sagittal section, the claustrum and the posterior part of the external capsule has been removed. And we can appreciate the superior longitudinal fasciculus that is coursing superficial to the optic radiation. It is superficial to the optic radiation and deep to the extreme and external capsule. So after insula and external and extreme capsules, we will encounter the superior longitudinal fasciculus, and in its depth, we will find optic radiations. Projection fibers, these are the fibers that pass up and down the neural axis. So above the thalamus, these fibers are arranged in a radiating pattern that is called a corner radiator. So this picture is only for the orientation that how the corner radiator is radiating before becoming the internal capsule. This is the corner radiator that becomes compact and become the internal capsule. And then as it goes down, it becomes the cerebral peduncle and it goes in the pons as pyramids. So discussing about the parts of the internal capsule, this is a thick mass of white matter that is bounded. Laterally, it has lentiform nucleus, and medially, it has caudate. Superiorly and inferiorly, it has thalamus. So it has 5 parts. It has anterior limb, a genu, a posterior limb. There are 2 other parts that are related to the lentiform nucleus. If it is just behind the lentiform nucleus, it is called the retrolentiform part. And if it is below the lentiform nucleus, it will be called as sublenticular part. So the anterior limb is located is laterally bounded by the lentiform nucleus and medially by the head of caudate. And it connects the frontal lobe with the pontine nuclei and anterior and medial thalamus. Whereas the posterior limb is bounded laterally by the lentiform nucleus again, but medially it has thalamus, and it connects the thalamus with the cortex, and corticospinal fibers to the motor nuclei of the upper and lower extremity runs through the posterior limb. The fibers to the arm are near the genu. The precentral gyrus is positioned superficial to the posterior limb. So the precentral motor strip is just superficial to the posterior limb of the internal capsule. Now, if we discuss genu, anterior and posterior limbs join at genu, and genu is the part where there are, other than thalamocortical fibers, there are corticobulbar fibers to the motor nuclei of the cranial nerves are present. So the retrolenticular fibers, some fibers of the internal capsule curve around the posterior edge of the lentiform, as we have explained previously, and referred to as the retrolenticular fibers. And some optic radiation fibers also pass through the retrolenticular fibers. So sublenticular fibers are important because it contains 2 type of fibers, which are auditory radiation fibers and optic radiations. So the auditory radiation fibers, they arise from the medial geniculate body, and they end in the superior temporal gyrus. And making the auditory radiation fibers, whereas the optic radiations, they originate from the lateral geniculate body and ends at the calcarine sulcus. So here, this is the sagittal section, midsection of brain, where we can see that the lentiform nucleus has been removed. Laterally, it lies lateral to the internal capsule, so it has been removed to expose the internal capsule. Here we can appreciate the anterior limb, which is between the caudate and lentiform nucleus. So if we remove this anterior limb, we will find the caudate nucleus. The posterior limb of the internal capsule is located lateral to the thalamus. So this is the thalamus, and posterior limb is going just lateral to it. And fibers of the internal capsule descend to form the cerebral peduncle, as we have seen in the schematic diagram, that these fibers will, and this is the genu, then the part between anterior and posterior limb is the genu, and these will descend as cerebral peduncle. So the retrolenticular part of the optic radiations here is removed, and to expose the tapetum, we will explain tapetum in the next slides, which separates the optic radiation from the ventricular wall. So tapetum is actually the part of carpus callosum, the posterior part of it, and it separates the optic radiations with the ventricular wall. It lies between them, and lateral epididymal wall of the atrium has been opened. So here the lateral wall of the atrium has been opened, and anterior commissure was transected and lateral part removed. So here we can appreciate that there is the anterior limb, the genu of the internal capsule, the posterior limb, and here is the atrium. These are the mid-optic radiations. We will discuss 3 types of other radiations, and anterior commissural fibers. These are present anterior to the internal capsule. This is a sublenticular part of the optic, and auditory radiation passes laterally and separated from the temporal horn by only septum. So here we can see that this is septum, which is found just behind the retrolenticular and auditory radiations. In front of it is the lateral geniculate body of the thalamus. The tapetum will separate the optic radiations and the lateral ventricle. So optic radiations are separated from the roof and lateral wall of the temporal horn and lateral atrial wall by only a thin layer of tapetal fibers, which we have seen and discussed. The fibers of the optic radiations are divided into anterior, middle, and posterior groups. So optic radiations is further divided into 3 parts. So this is the anterior fibers, middle fibers, and posterior fibers. The anterior fibers are called the Meier's loop, and these are the anti-optic radiations, which are called the Meier's loop, and they serve as the upper half of the visual field, lies just below the internal capsule. The middle fibers will serve as the macula, and the posterior fibers are responsible for the lower visual field. So in this picture, this is the same orientation in which the optic tract proceeds posteriorly towards the lateral geniculate body, and there are 3 bundles which we have discussed: the anterior optic radiations, the middle and posterior optic radiations that are found just below the internal capsule. These are some functions of internal capsule. So the anterior limb is associated with the frontal cortex and thalamus, and it is responsible for the complex cognitive functions, limbic connections. These are the areas of that contains the ascending cerebellar and basal ganglion connections to the motor regions of the cortex y thalamus. And on genu, we can see that there are corticobulbar tracts. Here, this is the supply of head, upper limb, trunk, and lower limb, and the posterior limb. Here we can see the ascending somatosensory fibers from the thalamus somatosensory cortex, and descending motor fibers from the motor cortex to the spinal cord nuclei. These are the auditory radiation to thalamus, the medial geniculate body, and the lateral geniculate body. So the medial is associated with the auditory radiations, and the visual radiations are associated with the lateral geniculate body. So commissural fibers, they are the fibers that interconnect the paired cerebral hemispheres. The largest fiber is the carpus callosum, that is located between the hemispheres in the floor of the longitudinal fissure and roof of the lateral ventricle. So it forms the roof of the lateral ventricle. It has 5 parts: 2 anterior parts, 1 body, and 2 posterior parts. This is the lateral ventricle, and we can appreciate that this is the rostrum genu. This is just a schematic diagram. We'll discuss the other pictures later on. The genu, the body, and the splenum is the little dilated part, and we also have a tapetum, which we have already discussed, but we'll see again. So the genu, it blends below the rostrum and continues quadrily as lamina terminalis. So lamina terminalis is actually the continuation of the genu, and it is the anterior portion of the third ventricle. Genu give rise to a larger fiber tract, fossa minor, which forms the anterior wall of the frontal horn. So in this section, what they have, it is a sagittal section, and they have removed the cingulum and the gray matter of cingulate gyrus. Here, they expose the radiating datary fibers of the carpus callosum. So genu anterior to the frontal horn, it gives rise to the fibers that interconnects between the two frontal horns, between the two frontal lobes. And posteriorly, it gives rise to a chunk of fibers that will form the forceps major that connects the two occipital lobes. So fibers passing through the genu of the carpus callosum forms the forceps minor, and anterior to the frontal horn of the large bundle passing posteriorly from the splenum forms the forceps major. So this is the splenum, and the fibers that are passing posterior to that will be the forceps major. And this splenum will form a prominence, the bulb of the carpus callosum in the medial wall of the atrium. So the posterior part of the carpus callosum is the splenum, and it forms a bulb, which is a prominence, and in the medial wall of the atrium. The genu blends posteriorly into the mid portion of the body. As we have discussed, the posterior part will form the fossa major, and that connects the two occipital lobes. This is just to have an idea how these fibers are present. So this is the forceps minor connecting the two frontal lobes, and this is the major uh, connecting fibers, which are the forceps major, connecting the two occipital lobes. So there's another tract, which is the posterior part of the carpus callosum, which is known as tapetum, that arises in the posterior part of the body of the splenum. And it actually sweeps inferior laterally to form the roof and lateral wall of atrium and temporal and occipital horns. So these set of fibers, they form the roof and lateral wall of the temporal and occipital horns. Um, so in this orientation, we can see that this is the sagittal cut. The quadrate nucleus here is removed, and to expose the fibers that are constituting the internal capsule. Here we can see the tapetum sweeps downwards to form the roof and lateral wall of the atrium and the temporal horn. So if we go beneath it, we will find the atrium, the temporal horn, and the tapetum actually separates the optic radiations which we have discussed previously from the temporal horn and atrium. So another type of commissural fibers are the anterior commissure fibers. These are the small bundle that crosses the midline in front of the columns of the fornix, which we will discuss later on. It forms part of the anterior wall of the third ventricle. So it is anterior to the third ventricle, and its shape is somewhat like a handlebars of the bicycle. It interconnects the olfactory structures and the temporal gyri of both sides. So its function is to connect the olfactory structures and temporal gyri on both sides. So this is the coronal section of the brain where it is at the level of the lamina terminalis is opened. This is the third ventricle, and we can see that anterior commissure is going just above the anterior third ventricle. Here above that is the fornix, and above that we can see the thalamus, lateral ventricles. So these are the anterior commissure fibers that are connecting the olfactory regions and temporal gyri of both sides. So this is a sagittal cut in which the external capsule has been removed to expose the putamen, and the anterior commissure interconnecting the temporal and septal areas. Here it is the anterior commissure just below the putamen. And the corner radiator spreads out the putamen, as we have already discussed about the corner radiator internal capsule. The So the third form of commissural fibers are the fornix. These are the main efferent pathways from the hippocampal formation, and it contains both the commissural and projection fibers. So these are C-shaped structures that extends from the hippocampus to the mammillary bodies anteriorly. So it has four parts. It has fimbria, the crus of the fornix, um, the body of the fornix, and the anterior columns or pillars of the fornix. So in this picture, we can see that the superior temporal gyrus has been removed. is the sagittal cut. The choroid plexus, which attaches along the choroid fissure, has been removed. The choroid plexus has also been removed. The outer edge of the choroid fissure is formed by the body of the fornix, and in the body of the ventricle, the crux of the fornix is formed in the atria, and the fimea is formed in the temporal lobe. Here we can appreciate that this is the choroid fissure, and here is the body of the fornix just below the lateral ventricle, and here it is the in between the medial to it is the thalamus, and here is the crus of the fornix, the fimea of the fornix, that is related to the temporal horn. So if we place in the same section, if we place a retractor in the thalamus and the crus of the fornix, we will be able to open the choroid fissure. Opening the choroid fissure in the body of the ventricle, we will expose the third ventricle, and opening the choroid fissure between the pulvinar and the crus of the fornix expose the quadrigeminal cistern over here, and opening the fissure between the lower surface of the thalamus and fimea of the fornix will expose us to the ambient cistern. This is the same orientation in which we can appreciate the body, the crus, the fimea of the fornix. So in this sagittal cut, they have removed the thalamus to expose the third ventricle. Here is the third ventricle, and the body, the crus, and fimea of the fornix forming the outer margin of the choroid fissure. So this would be the place of choroid fissure and have been preserved. Opening the choroid fissure in front of the crux will open into the quadrigeminal cisterns, as we have discussed, and the adjacent to the body of the fornix exposes to the third ventricle, and the fimea will lead us to the uh, ambient cistern. So here we can see the relation of the carpus callosum, the body of the lateral ventricle. Just below that is the body of the fornix, and below it we can see that there is foramen of Monro, third ventricle. Here is the pineal region. So this is the orientation of this picture. So talking about the fornix, it arises in the floor of the temporal horn. On the ventricular surface of the hippocampus. The fimea courses along the lateral edge of the lateral geniculate and separated from the geniculate body and optic radiation and by the choroid fissure. Posteriorly, the fimea blends into the crus of the fornix. We have seen that posteriorly crus will form the fimea of the fornix. So at the junction of the atrium and the body of the lateral ventricle, the paired crura meets to form the body of the fornix, which passes above the thalamus and below the septum pellucidum in the lower part of the medial wall of the body of the lateral ventricle. So at the anterior margin of the thalamus, the body of the fornix separates into two columns. So and at the anterior margin of the thalamus, the body will separate into two columns. Along the superior anterior margin of the foramen of Monro, and blends into the wall of the third ventricle as they pass behind the anterior commissure and descends to reach the medullary body. So anteriorly to the foramen Monro, they will blend, and behind the anterior commissure, they will descend down and forms the medullary body. So this is the memory body. So below, after coming So here the body and the crust are located deep to the lower part of the pre and post central gyra. The fimea is located deep to the lower part of the superior temporal gyrus. All of its parts are located deep to the posterior part of the insula. So in the
Can
body
you
of the
can you stop reading and kindly explain on the pictures that you have?
yes sir. So in this orientation, we can see that this is the sagittal cut, in which we can appreciate they have removed the thalamus, and here we can appreciate that this is the body of the fornix, and below it is the third ventricle. Above that is the carpus callosum, and this is the body of the lateral ventricle. This is the atria of lateral ventricle, and here we can see the crus, the fimbria of the fornix. So in this sagittal cut, much of the thalamus has been removed to expose the. This is the sagittal cut, and much of the thalamus has been removed to expose the body, the crus, and the fimbria of the fornix, forming the outer margin of the choroid fissure. So, as we have discussed, this is choroid fissure, body, crus, and fimbrial um, extension will go to the hippocampal head, and amygdala is just anterior to that. Now discussing the septum pellucidum, which stretches across the interval between the anterior part of the carpus callosum and the body of the fornix. So, septum pellucidum is the part which connects the carpus callosum to the body of the fornix. And if we see in this diagram, in the body of the lateral ventricle, the septum is attached to the body of the carpus callosum above and the body of the fornix below. So here we can see that this is the carpus callosum, this is the fornix body, and this is the septum pellucidum. So septum pellucidum disappears posteriorly, where the body of fornix meets the splenum. So at the part where the fornix meets the body of the fornix meets the splenum, the septum pellucidum is disappeared, and there can be a cavity that is called the cavum septum pellucidum in the midline between the lamina of the septum pellucidum. So in this picture, this is the coronal section. Sorry, this is the axial section of the um, okay, this is the coronal section of the brain, in which we can appreciate that there are two lateral ventricles that are separated by a septum pellucidum. Here we can appreciate foramen of Monro, and these are the basal ganglia and other external parts, claustrum, external and extreme capsule, and all those parts are just lateral to it. So this is just a schematic diagram to show what is the orientation. So this is the mid sagittal section of the human brain. Here we can see the carpus callosum, and this is the fornix. Between them is the septum pellucidum, the layer that is present between the both of the bodies, and as we go posteriorly, it disappears. And this is the splenum. We cannot appreciate that this structure is going backwards as well. So the there is a study that we use to see the white matter tracts. This is called the diffusion tensor imaging. So this is an MRI technique that uses anisotropic diffusion to estimate white matter organization of the brain. So the fiber tectography is a 3D construction technique to assess the neural tract using data collected by diffusion tensor imaging. So it has 3 types of color coding.
Dr. Adina, if you go back, what does the word anisotropic distribution means? What is the
Sir,
it basically
this is a
diffusion of water molecule?
this is a kind of a diffusion study, and
What is an isotopic diffusion?
I don't exactly know.
So what DTA does it tracks the diffusion of water molecules, and so it makes a pixel of it. Obviously, if you're mentioning a study and you're writing about it, you should read about it. So move forward. Tell us about the tracks.
I'll read up on that. So this is the 3D reconstruction, and this is the color coding of the tracks that we have discussed. The red color shows the transverse or the commissural fibers. The green is the association fibers, and the blue are the projection fibers up and down. So these are the color coding for this DTI. And the clinical application of this MRI would be if we want to see any uh, deformation or any problem in the white matter tract. For example, pre-surgical planning, if we want to do In Alzheimer's, it is used for early detection of the disease and focal cortical dysplasia or multiple sclerosis. It is used for the early identification of any of the white matter tract pathology. So this is the clinical application of DTI. Thank you. If there's any question, I would. Sir,
Uh,
if you want to ask anything.
Obviously, this is our first session online after a very long period, and I think one thing that every resident should see is that since it's online, the interaction kind of limits down. So the onus is on you guys to make sure that you explain it in a way that the resident understands. Obviously, this is the fitness stuff. It's not easy to understand. So I would urge the residents not to read through the slide, but actually to have diagrams or pictures of Rhoton to explain. So obviously, I urge you not to read the slides, but to make them understand. So obviously, you were a little fast, and I was trying to get Dr. Yasser on board as well as a co-host, but I couldn't. So obviously, the presentation was good, but I think the explanation could have been a little better. So the importance of tracks are because gray matter, it can reform this brain plasticity, but injuries to white matter are devastating, and a lot of work has been done on white matter tracks, and BTI is a very common imaging that a lot of people perform while doing the intimate surgery, especially on eloquent area, to decide what track or what approach they will take. And even people have studied white matter shift after brain tumor surgery, so there's a lot of work that's been done. But for us, so it's very important to understand just not what white matter is involved, but uh, uh, the white matter involvement or white matter information is I think more important than just knowing the gray matter, because as I said, gray matter in may not be as devastating as the white matter injury. So I think every resident should know about white matter tracks, and obviously it's difficult, but it's important to remember the common ones. And it's
Yes, sir.
so is there any question from the residents sitting in the room?
Sorry.
So if there's any question from the audience online, they can use the chat box. I know
There,
Dr. Yasser asked one question, and I know it's a trick question. So he asked, is Fornix a pure commercial fiber? Adina, is Fornix a pure commercial fiber?
Fornix is a mixture of both fibers, the projection and commercial both fibers.
Okay. Let's go back, read it up. And I think we'd like to conclude the session. Whoever is presenting next time should make sure that, as I said, that when we go online, the interaction between us becomes limited. So it's important that you guys have control over what you are presenting, and there has to be a balance between the speed and the level of understanding that you want to impart. Next time, I would also request you to record these presentations, that so that we can put them online. If there's no question, then I'd like to conclude the session.
Okay, sir.
Thank you, everyone, for joining online and those sitting in the seminar room at the National Hospital. Thank you for your
Thank you, sir.
presentation.
Thank you.
Agitate with me even you have to या तो हम लेंगे localize करके and then we remove the itis and we do it without itis. We use that itis in bacteria.